Now we move on to the next speaker, Professor Hsing. So Professor Hsieh, he is the director, distinguished investigator, and the director of the Institute of Biotechnology and Pharmaceutical Research in NHRI. I want to, because the Professor Hsieh, he is really one person in NHRI very respectful, because in one hand, he do a lot of the very research achievements, so win a lot of the research award. Just recently, is an award from the Education Ministry of Education, the 68th Annual Academic Award. It's not very easy to get the award. So he got a lot of medal in one pocket of his pants. But another side, he is also earned a lot of money from his, because he developed a lot of the drug and translate to the clinic and also license to the industry. So he got a lot of the license fees. So let's, today he's gonna give us a talk about targeting KRAS mutation in cancer from the medicine, chemistry strategies and the insight. Let's welcome Professor Hsieh.
Okay, so thank you, Dr. Chang. Good afternoon, everyone. It's my great honor. I don't know how to say it's my great honor to share with you today the recent progress in targeting KRAS mutation in cancer and medicinal chemistry insight that made this breakthrough possible. For over 4 decades, the KRAS was the level as the undruggable, this played the central role as the cancer driver. Just Dr. Hsieh talked before. All the early attempt to inhibit it failed. However, the advance in the structural biology and the computation in modeling and the covalent and drug design have turned what was once impossible to reality. Today, I will walk you through how the chemists and the biologists have worked together to transform the KRAS from the symbol of the frustration into the remarkable success, the story in oncology. Before I talk to my talk, I should admit that I personally don't have any research regarding the RAS. Okay, that's why I say I don't know why I stand like here, because I didn't do any RAS research. But however, in my NHI, we have you can see 8 of the 10 and development candidate have been licensed to the industry partner. 8 of the 10 are the cancer. And then currently the 3 in the preclinical or 4 in the phase 1. And then finally we have the 1 diabetes drug was approved this year. Only one of the 5, which is currently searching for license, the 728 is the kinase inhibitor targeting to the MYC-driven cancer for cancer. Okay, so there's only one relationship with today the topic. Another, we have currently we have the 4 pipeline, and one of the MYC-RAS inhibitor is related to the today the topic. So at beginning, I think the Hsieh Hui should be speaking for this one. But the Director Cha told me, hey, you should talk about from the medicinal chemistry of the view. So that's I give it the 5 parts. The first part is the biology. Second is the breakthrough, the discovery, the KRAS off, the G12C inhibitor, and the third is the target other KRAS mutation in G12D and the G12V, and the fourth is the target the KRAS on or KRAS on off or pan-KRAS molecular glue degrader, and finally is the future perspective. So the next maybe 11 minutes is only literature review, okay? It's no personal research. Okay, so for this slide, I already Dr. Hsieh, but people should know that in 78, Edward Sorensen is from the Merck. He identified the KRAS. So the K is the Christine, okay? So as you know, the KRAS is one of the most frequent mutant oncology. It's about 25% of the cancer. In particular, the pancreatic cancer is 88%, colorectal is 44%, and lung cancer is 32%. So the huge unmet clinical need, 1 million new cases per year. And a driver is control the growth. As you can see, it's a different mutation in lung cancer. It's a G12C, and in CRC it's the G12D and the G12V. For pancreatic cancer, it's the G12D. So I think I saw before that Dr. Hsieh will talk about cancer biology, but I'm not. But as you can see, if you develop the KRAS off, it's the block the exchange of GTP and the GTP form of the KRAS. But if you block the KRAS on inhibitor, is disrupt the protein-protein interaction of the KRAS GTP and the downstream effort. You should know on and off, okay? So RAS is at the beginning we think, okay, tyrosine kinase inhibitor brought the ATP binding site. So can the same approach work for the GTP binding site? It's quite logical, right? So because in 2001, immediately Gleovac was approval by the FDA. But in the past 25 years, it's already 96 the small molecule, the TKI kinase inhibitor was approval by the FDA, but only two molecule or two KRAS molecule approved in the recent 5 years. So the reason is very simple. ATP binding affinity is around low micromolar, but GTP binding affinity is a picomolar. Okay, so it's very difficult to compete in the it's very difficult to make it extremely difficult for small molecule to compete. And the second, RAS is spiral in shape, so it's a smooth surface. There is no deep binding pocket and a multiple variant compressed network. So that's 40 years the failed attempt. So the breakthrough strategy, the key turning point in the discovery of the switch to pocket. As you know, the Kevin Suke in UCSF, he found out in structural biology the one mutation called G12C is around on the switch 2. If they can find the compound, and then they can bind to the this side and the locking the mutant protein in the inactive form. So we call off, right? So at the beginning, I don't show a lot of chemical. I did show the chemical structure, but I don't go the detail. So I can at the beginning I think, okay, cysteine, they can attack the disulfide bond. Easy, right? But the affinity is low until they found out they can build up like introduce the microreceptor. So basically learn from the success of fattening with the amide type microreceptor approved by the NSCLC in 2013. So you can see it here. The reason is you can see Kevin Shuke discovered this compound in 2013. At that time, the 2013 fattening is the first drug to use we call the microreceptor. Okay, they can form the covalent bond by the cysteine, and after that is the 3 BTK inhibitor use the amide type the microreceptor, and the 3 more EGFR, and then 1 FGFR, and then 2 KRAS 12C. Okay. So the Kevin Suket at that time found the compound, and then I think he got a spin-off company called Wellspring Bioscience. But at that time, the potency is only 1.6 micromolar. So after optimization, well, still is around 150 nanomolar. For ECP is 1 to 3 micromolar. But after the Amgen and the Mirati, they have a very strong medicinal chemistry, so they discovered these 2 compounds. One sotorasib is around like 8 nanomolar. For Mirati is around 0.6 nanomolar. So finally, both compounds was approval by the FDA. Okay, so G12C is covalent inhibitor, and the scaffold is basically we call a 6-6 membrane, and then they can introduce the 3 pharmacophore. So currently it's around at least 7 compound is on the clinical trial. A lot of the big pharma like the Novartis, AstraZeneca, Lilly, Genentech, Boehringer Ingelheim, and Merck to develop their own G12C inhibitor, and Inventive Bio from China. They also have their own G12C inhibitor. So I just should point out they can use the microcyclic to make it more rigid, to increase the bioavailability and potency. You can see this and Eli Lilly, and the Merck is also have the microcyclic molecule. So the key SAR insight for the G12C is the switch to Parky occupation, very good microreceptor as the electrophile, and then lipid fertility optimization and the conformation selectivity, and then finally is the PK and the CMS balance. You can see as the example, AZD is the limited CMS penetration, but remove this functional group, they can have a higher CNS exposure. So again, the KRAS mutation, the key turning point is learn from the success of the G12C inhibitor. As you can see, the G12D is very important. So for the G12C, it's formed a covalent bond, so it can gain the energy about 100 kJ per mole. But for ionic, it's around 60 kJ per mole. So you need to have the more interaction. So currently, I should use the 1133 as an example. Mirati developed their compound based on the G12C. So they just change from this side to the amine side. But however, amine-based hydrogen bond interaction, and they only can get around 3% oral viability. So finally, in 2025, BMS stopped the development of this compound due to the PK data is highly variable and suboptimal. Okay, so PK is quite important. Okay, and then we'll move the KRAS on and off. As 50% the patient require the resistance, and off the inhibitor only suppress the GTP bond, but the on inhibitor is disrupt the protein-protein interaction. So currently you can see G12C on is Merck in phase 3, G12C on off in phase 1B, and then now a lot of company focus on the G12D on and off the inhibitor, at least have the 5, 2 from the China company. And then we move to the KRAS. If you can bind the 2 of the more KRAS side, it's more to make a more broad inhibition. For example, engine 410 is as the example, and at least the level another two drugs from the China company. So I don't go the detail, but you can see they start them 3 micromolar. Okay, and then they improve the 1,004 for the enzyme activity. For cell-based, is the 10,004. So you mean so their chemistry is still very, very good. So now the 410 is the bind the allosteric the pocket, and then it have the selective the scaffold, and then with the microcyclic the scaffold, and then they have the dual on and off, and then have the higher selectivity for HRAS and the MRAS, and you can see the data. Then we move the new modality called tri-complex inhibitor as the molecule group. So I don't go the detail. Is the tri-complex utilize the intramolecule chaperone called cyclophilin A as the molecule group to form the either non-covalent or covalent inhibitor tri-complex to further to block RAS-MEK signal pathway. Is the animal data is pretty good. Okay, so currently they have the 2 from the Revolution Medicine. One is the non-covalent, and the clinical is already in the phase 3. So Dr. Lin Jiaqi will talk about the clinical trial data later on. And then the one covalent for G12C is in the phase 1 and phase 1B. And also is the one compound from the Jungai Roche, but they are discontinued this year. So, and then also for the tri-complex for molecule group. So you can see the Revolution level the 2 compound. One is the 9805 is in the phase 1, phase 1B, and G12V is they also have the 1 compound for G12B. As you can see, the RNC 9805 is really display high quality the drug-like property, and then is highly selective for crossing the KRAS the G12D, only inhibited G12D. And then the drive the regression of the KRAS G12D in the xenograft. So the data is very good. And then also the also is the animal data, so you can drive the deep durable or regression across the driver G12D the cancer model in vivo. So finally, another new modality called KRAS the degrader. So you can see here, if you use the traditional inhibitor, they show like the oral twice a day. But if you use the degrader, you only need twice a week. And then the response is pretty good. Currently, they have the 4 degrader, 2 for G12C, 1 for the G12D, and 1 for pan-KRAS. So finally, overall, the advanced KRAS targeting, as you can see, already have the 2 G12C was approval. Okay, so what you can do? A lot of the company go follow. Currently, they have the 7 7 compound for the G12C in the phase 3 clinical trial, but I don't know they can beat the recipe. But it's one called pan-KRAS. I think Dr. Lin will talk about the clinical trial. And then 5 compound in G12D, and the 5 compound for G12C, and then 2 compound for pan-KRAS in phase 1, and the 4 pan-KRAS in the phase 1. Okay, for the future prospect, resistance is the obvious, okay, but it can be delayed through the smarter design and the combination. So the expand beyond the G12C. Before the G12C, people didn't know that if you can block the switch pocket 2 to block the whole pathway. So now they know the G12C and then go the G12D and pan-KRAS. And then how to overcome the resistance is by rational combination. And then they also developed the novel modality called protein or molecular glue. And then people, the AI-driven design to accelerate the drug discovery. And finally, the personal medicine is the biomarker guiding the therapy. So the next decade of the KRAS should be shaped by the AI and the new modality. So it is the AI capacity. So I don't go the detail, but you can know the AI is quite important. So final take-home message, the KRAS is no longer undruggable. G12C, the inhibitor, make a new era. Medicinal chemistry drive the precision oncology, okay? By the SAR and the structural design are the key. The future is innovation, integration, new modality, combination, AI, and personal and medicine. So KRAS the jury. Everybody face the challenge. Can you find something to break through? And then finally, you will get the success. So one more advertisement, okay? Since I'm the director of the Institute of the Biotech and the Pharmaceutical Research, we welcome everybody to have the good the topic, so you can join our group from the target to the new drug. And the next year we have the 3 centers supported by the NHI. One is called VMIC, is the small molecule platform to optimize the compound and bridging the preclinical study. The second is called KMIC, is the molecular platform for protect messenger RNA, siRNA, LNP, and the covalent inhibitor. The third is called AMIC, is the AI-driven drug development, and the DMPK, the system for efficient innovation. So finally, once again, thank you, Dr. Cha, give me this presentation, because I collect all of this information in my past 2 months. Okay, thank you for your attention.
Okay, thank you. One short question? Any question? Okay.
Okay, just one question. Why always in the G12 region? You change the drug treated there, and then the mutation is still in that area. But what is the reason for that?
I think they need a combination. Okay, so when they treat the KRAS, they cause.
Why the mutation always hit the G12 area? Yeah. So the mutation, but maybe we can, David. Yeah. You any suggestion? Church, Dr. Church? You do you know? So that's $1 million question. Okay, so we don't have time for the further discussion, but I have to remind you why you today here, because our president Hsing-Pang here, because you got a new assignment, because none of these currently show very promising for pancreatic cancer. So that's your mission, to have the new KRAS inhibitor. Okay?
Okay. Right. Thank you.
Thank you.